Welcome to Infectious Disease Epidemiology. I'm Peter Millard, and my co-facilitator for this course is Amy Kinner. The course actually has two mottos. The first motto is a fun course, and the second is not much math. This is a higher level course. You have the basic skills of epidemiology that you learned from Amy in Principles of Epidemiology, and I'm not going to repeat a great deal of that material. In this course, you're going to be expected to synthesize all the information and the skills you have from Principles of Epidemiology in order to be able to demonstrate your ability to think critically and not to just regurgitate facts. In that respect, definitions are incredibly important in any technical field, and especially important, I think, in infectious disease epidemiology, which has a lot of fairly specialized definitions. But my assumption is that you'll learn them through the readings. The principal textbook has a lot of the definitions, plus there's an article that we've given you to read that deals specifically with defining terms in infectious disease epidemiology. The course emphasizes the great challenge of critiquing scientific evidence to established published health practices. The key skills that you're going to be practicing in this course are working in a team, writing, expressing your views about the scientific evidence that you encounter, critiquing the works of others, and responding to the critiques of your work. Therefore, clear communication skills are something that you'll be able to develop in this course. And you'll spend your time most productively by reading and studying, communicating and working together as a group with your team members, and working on your projects. Lectures are less important in this course, and I won't cover a great deal of factual material, but I will emphasize a few of the basic skills that we're going to need. I say that this is a course with not much math because there really isn't. We do have a quiz in which we're going to ask you to, an open book quiz in which we're going to ask you to demonstrate some of the knowledge that you gained from Principles of Epidemiology. Otherwise, there are 5 group assignments. There are online forum discussions, which you're used to doing. There are 2 epi-info exercises and a final paper, which will also be done in groups of 4. I want to start with a quote from 1967. The U.S. Surgeon General, Dr. William Stewart, said in 1967, "It's time to close the book on infectious diseases, declare the war against pestilence won, and shift national resources to such chronic problems as cancer and heart disease." Now, from today's perspective, that might be seen to be a really stupid statement to make, but Stewart's not the only one that said stupid things, and in fact, that was 1967. But in around 2006, the head of the National Cancer Institute, who was a urologist, Dr. Andrew von Eschenbach, who was appointed by George W. Bush, said, "The goal of the National Cancer Institute is to eliminate the suffering and death due to cancer by 2015. So we have people making silly statements today, just as we did in 1967. On the other hand, the Director General of the WHO, Dr. Nakajima, said in 1996, "We are standing on the brink of a global crisis in infectious diseases. No country is safe from them. No country can any longer afford to ignore their threat." And I think almost 20 years later, that statement, the truth of that statement is very obvious, and we're meeting new challenges from infectious diseases on a regular basis in the world, and I'm sure that's going to continue. I'd like to start off with a little bit of a story about Antoine Lavoisier, who lived in the 1700s in France. He was considered the founder of modern chemistry. He was the person who determined that water is made up of hydrogen and oxygen. And Lavoisier, like many scientists of his time, wore many different hats. And one of the jobs that he did was he worked for the French government as a tax collector, which wasn't a very good idea at the time of the French Revolution. But Lavoisier, before he ran into that problem, was a very well-known scientist in France and very well respected. And he was appointed to be on a committee of the French National Academy of Sciences. And that committee, another member of that committee, by the way, was Dr. Guillotine, who developed what they thought at the time was a much more humane way of executing prisoners. And a side note to the story is that Lavoisier himself ended up being guillotined during the French Revolution for his activities working for the government. But at any rate, Guillotine and Lavoisier were members of the committee of the French Academy, National Academy of Sciences, who were asked to judge the efficacy of a treatment which was widely known at that time, which was developed by a doctor named Dr. Mesmer. You may have heard the word mesmerize that comes from that. But Dr. Mesmer had a theory of animal magnetism. And in his theory, if a group of persons sat around in a group and put the ill person in the middle, and they concentrated their animal magnetism on that person, they could cure him from diseases. So it was Lavoisier and Guillotine, among others, job to evaluate Mesmer's claims and give an opinion about whether this was an effective way of treatment. They didn't think much of Mesmer's treatment, in fact, but afterwards, in 1784, Lavoisier made a statement which I think is extremely true today, and I think may be really applicable to this course. And Lavoisier said the act of drawing conclusions from experiments and observations consists in evaluating the probabilities and estimating whether they are large enough to constitute a proof. He said this type of calculation is more difficult than is generally appreciated. It requires great knowledge and is beyond the capacities of most men. I added even women. The evaluation of probabilities is especially difficult in medicine. When one administers remedies, it is incredibly hard to distinguish what to attribute to nature and what to medicine. That's a very, very true statement even today, but I think that where I disagree with Lavoisier on this statement, is that he talks about probabilities. And I think when we evaluate scientific evidence, it's not the probabilities, I think, that are especially difficult. I think that what is especially difficult is evaluating the validity of claims. And you remember, let me just review for a second the difference between probabilities and validity. In order to do that, we'll talk about the difference between epidemiology and statistics. In epidemiology, the primary focus is on assessing the validity of scientific evidence. So we ask the question, do the data reflect reality? And largely, we use judgment in epidemiology in order to determine the validity of statements. So that subject matter and expertise in the subject becomes incredibly important in epidemiology. Whereas I think that the role of statistics has been pretty well defined, the role of probability. We ask in statistics, what is the role of random error in the data and how precise is the estimate? So that we use p-values and confidence intervals in order to estimate the effect. So when we talk about validity, what we do in epidemiology, it means accuracy or truth, and a valid study is one that gives a true or unbiased result. Whereas precision, again, relates to statistics, and that refers to reproducibility or reliability, the degree to which repeated measurements of a stable phenomenon get similar results. So, therefore, a study with few subjects would be imprecise. So validity of a study is influenced by the type of study design used. Of course, the type of study design has to reflect the question being asked. Randomized controlled trials are not always desirable or possible, but in general, when we talk about treatments or preventive interventions, it's true to say that randomized controlled trials are often more valid than cohort studies, which are in themselves more valid than case control studies. But how well a study is conducted, the quality control of the study, is also incredibly important. So in addition to looking at study design in this course, you'll also be evaluating the conduct of studies. Whereas precision is influenced by the inherent variability in the data, how the data are collected, and the sample size. But remember that bigger is not always better. Some of the biggest blunders in medicine and public health have been because of large studies that were not valid. They were biased. The most current example that comes to mind is the misadventure with postmenopausal hormone replacement therapy, which had very large studies of the type of cohort, that showed that on the balance, hormone replacement therapy was beneficial for postmenopausal women in terms of less heart disease, less Alzheimer's disease, etc. However, when randomized controlled trials were done, it was understood that the net balance of hormone replacement therapy is actually negative, that it actually increases the rate of heart attacks, strokes, pulmonary embolism, and breast cancer. So the problem wasn't the size of the studies. The problem was the validity of the studies. The investigators were using the fundamentally wrong study design in order to evaluate a treatment. So remember that a small study may be valid and a large study may be biased. And I think we'll need to keep that in mind during our work in this course. So the assumptions of epidemiology, of course, disease doesn't occur at random. It has risk factors that are measurable. Diseases have causal and preventive factors that can be identified and changed to improve human health. And the social causes of disease are at least as important as the biological causes. And I think that's one of the emphases of this course. Let's just talk a minute about the web of causation and look at the simplified version of epidemiology that we usually present in introductory courses. And think for a minute about how that simplified relationship might not be quite accurate. The next 17 slides I borrowed from Dr. Thomas Songer from the Aga Khan University, who also, you'll notice, attributes some of the slides to others as well. But let's look. The basic question in analytic epidemiology is, are exposure and disease linked? Okay? And when we examine the link between exposure and disease, we have to be very specific about what exposure we're talking about. We have to define very carefully the exposed, what are the potential health effects of that exposure, and what approach will we take in order to study the relationship between the exposure and the effect. So a good exposure variable should have an impact on health. It must be measurable. You need to be able to differentiate populations between those who are exposed and unexposed. You need to generate testable hypotheses, and ultimately, the idea is that it will help to prevent or control disease. And the qualities of a disease which make it worthwhile to investigate are that the disease must be important in terms of the number of individuals it affects and the type of individuals it affects as well. Remember the quality adjusted life years from principles of epidemiology. Diseases that affect younger people are more significant epidemiologically than diseases which affect older people. The disease must be important in terms of the types of population it affects, and the disease is important in terms of its causal pathway or risk characteristics. So we ask, is there an association between exposure and disease? And finally, once we ascertain that there is an exposure, the difficult part becomes, is the association real or causal? The big picture is that we look for links in order to be able to intervene and prevent disease. And we need to think about the basic definition of a cause. Therefore, exposure must lead to new cases of disease. And if we remove the exposure, most cases don't occur. On a population basis, this means that an increase in the level of a causal factor will be accompanied by an increase in the incidence of disease, and vice versa. If we eliminate or reduce the causal factor, the frequency of disease will decline. In infectious disease epidemiology, investigations are undertaken to demonstrate that association. But unfortunately, things are not so simple. The cause or etiology of disease is often difficult to determine. We don't know what causes some of the major cancers, for example, breast cancer, colon cancer, prostate cancer. We know that there are risk factors for those diseases, but it's very complex to try and determine in a simplistic or reductionist manner what is the cause of any one disease. Many exposures cause more than one outcome, and outcomes may be due to multiple exposures or continual exposure over time or exposure only in a particular context. And of course, causes may differ by individual depending on the genetic makeup and other characteristics of the individual. Of course, epidemiology doesn't determine the cause of a disease for any given individual. It determines the relationship or association between a given exposure and a frequency of disease in populations, and we infer causation based upon the association and several other factors. Association implies that exposure might cause a disease, and most often we design interventions based on associations. And remember the all-important confounding factors, those additional factors which are associated both with exposure and disease, which need to be accounted for in order to measure that association. When we talk about the web of causation, however, there's no single cause for any disease. The causes of disease are interacting, and the web of causation illustrates the interconnectedness of possible causes, so that the simple relationship between exposure to disease is in practice not very helpful, because every disease is caused by a web of causation, such as the social organization, individual behavior, the environment, the workplace, genetics, the particular microbes or organisms that are involved in infectious disease epidemiology, the phenotype of the individual, and also many unknown factors. So that Hill's criteria for causal inference, which you're familiar with, maybe aren't quite as useful when we talk about the actual web of causation, because it's a very reductionist way of looking for a single exposure causing a singular disease. And unfortunately, most of those relationships were determined long ago, and the difficult questions today are much more complex than a single exposure disease relationship. Ultimately, though, we do need to weigh the weaknesses in the data and other explanations against the science and the results of causal models, so that causal relationships are just the top of the pyramid of relationships, associations that occur by chance alone, hence the importance of statistics. Spurious or artifactual or confounded relationships as well. So that the social causes of disease make, I think, any study of epidemiology, and particularly infectious disease epidemiology, much more difficult than, unfortunately, what we learn in basic skills of epidemiology. Diseases have biological causes, but they also have social causes: poverty, inequality, unemployment, poor nutrition, exploitation, abuse, war, to name just a few. And the social causes create the biological causes. For example, HIV/AIDS is caused by a virus. The virus is transmitted sexually through blood or through blood contamination. However, the transmission of HIV, and also determine largely who actually gets sick and how long it takes to sick when they get HIV, are related to poverty, which drives women to sex work, inequality of women, which makes them unable to negotiate safe sex, unemployment, which forces men to go to distant places. For example, in sub-Saharan Africa, many men go to work in the mines in South Africa. They're separated from their families, and end up inevitably bringing back sexually transmitted diseases to their communities. The lack of adequate medical care, both for treating sexually transmitted infections and, of course, for treating HIV infection and preventing the transmission to others through treatment of HIV infection. The root causes of tuberculosis, again, are very complex. What causes someone to be exposed to the TB bacteria in the first place? If exposed, what causes him or her to be infected by it? And once infected, why do some people get sick from it and others recover fully? And then why do some people then spread the infection to others? So these are not easy questions, and I think they're at the forefront of the work that you'll be doing in this course. Of course, we've always had severe, even fatal sexually transmitted diseases in the world, and this is a poster from France in 1884, Protect Yourself Against Syphilis. It was only a few years from the development of antibiotics in the 1940s when we could treat syphilis and therefore had no very common fatal sexually transmitted disease, although, of course, HPV is also a fatal, potentially fatal sexually transmitted disease, and others, until HIV came along in the 1970s and really changed the whole picture. I'd like to talk a little bit, again, one of the basic things we learn in epidemiology is about the epidemiologic transition. And I hope that you'll see during this course that the epidemiologic transition is a myth for most of the world's population. The idea that infectious diseases dropped, the idea that the Surgeon General said that we can close the book on infectious diseases and that we need to concentrate on non-communicable diseases because they've become much more important. Of course, that was never really true in the huge majority of the world's population, and it's still not true today in most of the world's population. Infectious diseases always was and is now and probably always will be a key cause of morbidity and mortality among the world's population. So if we look, for example, at the cause of death in Maputo in the 1990s, the leading causes of death were HIV/AIDS, death of the newborn or mother, malaria, diarrhea, tuberculosis, pneumonia, all except for the perinatal disorders, all infectious diseases, although preeclampsia, which is a major cause of maternal death, may also be an infectious disease. And if we look at the rates of maternal mortality in the world, we can see what huge differences there are. Mozambique, where I worked, having a maternal mortality of over 1,000 per 100,000 women at the top of the list. And then if we look at the bottom of the list, at Grenada, Greece, and so forth, we've got rates of 1 per 100,000 women maternal mortality, so 1,000 times less than the maternal mortality in a place like Mozambique. And of course, there are huge differences in mortality as well. Overall mortality in a place like Iceland, where the overall life expectancy is 79 years. That's both men and women combined, compared to Zimbabwe, where the life expectancy is about 38 years. In fact, all the countries with the worst life expectancy are countries that are severely affected by HIV/AIDS or severely affected by wars in the case of West Africa. So that you can see also that there are huge differences in the prevalence among people 15 to 24 years of age by sex in selected countries. That in a country like Swaziland, also notice the red bars are females, the gray bars are males. The prevalence among women in Swaziland during that time period was over 20%, whereas in other countries, even African countries like Senegal, the rate was well under 2%. So huge differences in infectious diseases, also geographically. Again, and we'll talk about this in more detail when we talk specifically about HIV and AIDS, but the prevalence, we think of HIV as being an African problem, but really it's a problem everywhere, but it's not specifically an African problem. It's specifically a problem of southern and partly eastern Africa. If we look at the green lines here, which represents Swaziland, we can see that the HIV prevalence among pregnant women during this period attending prenatal clinics was over 40%. And if we look at the x-axis, the rates in southern Africa run between 10 and 40%. Whereas if we look at East Africa, not only, I think the striking thing here is how fast the rates are declining, but at the current time, the rates are between 5 and 10%. And in West Africa, the rates are usually between 1 and 5%. And again, if you look at Senegal has about the same rate of HIV infection as the United States. So huge changes or differences in mortality and prevalence of infectious diseases, even within the less developed countries. So you're well familiar with the epidemiological triad, which talks about the relationship between the host, the agent, and the environment. And this is the sort of analysis that we'll be thinking of in this course, that the environment plays such a huge role as well as the host and the agent. And in fact, the health determinants are probably more strictly related to environmental conditions than individual conditions. If you look at this puzzle of health determinants, income and social status, social support networks, education, physical environments, health services, etc., biology and genetic are actually just one piece of that large puzzle of health determinants. I'd like to switch topics a bit and just review for a second, I think, about John Snow in London in the 1840s and 1850s, who was the first infectious disease epidemiologist who investigated outbreaks of cholera in London in that time period. And cholera being a disease that causes fulminant diarrhea that can lead to death in a very short period of time, of course, continues to be a serious problem in the world today. So we have a great deal of scientific evidence about the cause and the prevention of cholera, and yet, again, another one of the themes in this course is that in spite of scientific knowledge, that it's not enough to have scientific knowledge to change people's health status, but that culture and politics become incredibly important. So cholera at that time was thought to be caused by miasma or bad air. The word malaria, of course, meaning bad air as well. And so most diseases were thought of at that time because the smells were so bad in the cities because of all the sewage that was around. This was thought to be the cause of many of the diseases. But Snow, of course, had a different hypothesis, that it was actually dirty drinking water that was causing the cholera. So John Snow studied the outbreak. He made an epidemic curve showing the attacks and the deaths having a slightly different pattern, the attacks leading the deaths by a couple of days. And he made a map of all the cases of cholera around the Golden Square area of London. This was in 1854, and noted the geographic clustering of cases around the Broad Street pump, which gave him the hypothesis, allowed him to test the hypothesis that different water companies, which took water from different areas of the Thames, might actually have different rates of cholera that could help explain the differences. And so he used some of the same relationships that we use today. He calculated the deaths per 10,000 houses using water supply from the Southwark Company, which supplied water to the Broad Street pump, the Lambeth Company, and then comparing it to the rest of London. And you could actually calculate very easily a relative risk. The relative risk comparing Southwark to the Lambeth Company was about 9. So the death rate per 10,000 houses was about 9 times as high in those who received water from the Southwark Company compared to the Lambeth Company. Again, the epidemic curve and Snow removed the handle from the Broad Street pump on September 8th. And at that point, the attacks diminished and finally not resolved completely, but were much fewer than the prior. So the reason why Snow is so important was because, of course, it was the first real epidemic investigation, but it also was accompanied by a cure of the problem. He counted cases, he used rates, he used geographic analysis, and he made an epidemic curve. So he used many of the methods that we use today and that are very important in outbreak investigation, and we'll have a chance to practice our skills and learn how to do an outbreak investigation during this course as well. James Lind, on the other hand, who came significantly before John Snow, was the first person to conduct a clinical trial. He was a Scottish medical doctor, and as you know, scurvy is a vitamin C deficiency disease that's caused by extensive soft tissue changes, bleeding of the gums, and susceptibility to infection, such that scurvy in the 1700s caused more deaths than military conflict. And in Anson's circumnavigation of the globe, for example, in 1740, 1,400 out of the 1,900 men died of scurvy. And so James Lind took 12 patients. This was a clinical trial. It wasn't randomized, but he did make a case definition. He said the cases were as similar as I could have them because he wanted to be sure that they were all scurvy cases and not other cases of other diseases that were masking as scurvy. So 2 of them, he gave a quart of cider a day, and so he gave different treatments to each group. And he said that of the two men who took oranges and lemons, only they improved, the other ones didn't. And so in 1754, Lind published a treatise of the scurvy, which was widely ignored, which is, I think, one of the lessons of this course, that scientific knowledge is not enough to really make changes. And it was about 40 years later, after other mariners noted the same relationship, that before citrus was finally used on ships to prevent scurvy, and British sailors from that day on were known as limeys because limes kept a little longer and were kept on ships to prevent scurvy. But of course, many indigenous populations, for example, the Native Americans, didn't have scurvy because they themselves learned that if you boil pine needles and drink the tea, that also contains enough vitamin C to prevent scurvy. So the British could have also learned from many of the native populations. The last story that I want to relate to you is that of Dr. Ignaz Semmelweis. Semmelweis was a Hungarian doctor who worked in the first obstetrical clinic in Vienna in the 1860s. And during his work, he noted that maternal mortality, primarily from infectious causes, was 11.4%, and that exceeded by far the mortality in the second obstetrical clinic, where the laboring women were attended by midwives. That is, the relative risk was about 4. The rate was 4 times as high in the first obstetrical clinic compared to the second. Semmelweis performed and published various studies to identify the causes of the mortality differences, and his conclusion was that the main difference was that the doctors dissected cadavers and didn't wash their hands before then going to the labor room to deliver babies, and therefore were infecting the laboring women. Semmelweis made 3 recommendations. He recommended the isolation of infected cases, hand washing before attending patients, and boiling of surgical instruments. In 1847, Semmelweis fixed to the wall of the door of the obstetrical unit a sign that said, "Starting today, every doctor is obligated to wash hands with a solution of chloric acid in the basin at the entrance before entering the labor ward." And here's an artist's rendition of doctors washing their hands prior to attending patients. Unfortunately, Semmelweis's findings were not accepted by the medical community. In fact, they fought them wholeheartedly, and he was judged insane by the Medical Society of Budapest, which pushed for his institutionalization. He was institutionalized in an insane asylum in 1865 and died 2 weeks later as a result of beatings by the guards. It was many years after Semmelweis's death that Pasteur's work on the germ theory led to the acceptance of hand washing and antisepsis. The conclusions that I draw from these scenarios is that scientific discovery by itself is not enough, that politics and culture determine the dissemination and acceptance of scientific studies, and therefore that clear, cogent communication skills are essential for public health practitioners. Think, for example, about the debate about global climate change. The scientific evidence for climate change has been well established, but financial interests, particularly the fossil fuel industry, has had a major campaign to prevent the scientific evidence from being accepted by the population, to prevent real change from occurring. They've done this in a similar manner to that which the tobacco industry used very successfully for many years to combat the idea that smoking was bad for your health and to prevent more regulation of the industry. So in conclusion, public health is multidisciplinary. Teachers, community leaders, engineers, economists are incredibly important. That it's multi-sectoral. Think for a moment about the severe obesity problems we have in the United States and other developed countries. We're never going to solve these problems through a medical model. We need to look at the food system, the work system, the entertainment system, the transportation system. Again, it's a good example of a problem where there's a web of causation which is very complex and very difficult to sort out. Public health focuses on equity and socioeconomic determinants of disease, justice, meeting the needs of those with the greatest need, and addressing the root causes of ill health. Public health involves collective responsibility. We have a responsibility for our own health and that of others. It involves the empowerment of and partnership with communities. It emphasizes prevention over treatment, and decisions are made on the basis of scientific evidence, not opinion. So the public health practitioners are advocates for communities and agents for social change. If you want to be an agent for social change, you have to have a very good grasp of the scientific evidence and also be able to communicate that to the public. So these are the skills we're trying to get forth in this course. This is the end of the first lecture. As I mentioned, the lectures are not really the most important aspect of this course. You have assigned readings. I don't expect you to only read the assigned readings, but also read as extensively as you can in the areas that you're researching. I will only be available by email for the first 2 weeks of the course. Amy will be available by the general means of communication, but I encourage you, if you have any questions, to read the syllabus carefully first. If you have any questions or concerns that you need answered, please communicate with us directly. If you have questions, there's a pretty good chance that other people will have the same questions, so please don't hesitate to ask. All right, well, that's all I have for today, and take care.